In this session, we will cover phylogeographic models to elucidate the processes underlying the genetic diversity of populations in space and time. Phylogeography is the study of the historical processes that may be responsible for the past to present geographic distribution of lineages. Locations may be continuous, as in longitude and latitude, or discrete, like cities or countries. Phylogeographic patterns result from a three-way interaction among the rates of transmission, spatial movement, and viral evolution. Although some viruses can persist in the environment for more than 1 month, like rotaviruses or picornaviruses, many others must be directly transmitted. Thus, their dissemination is strongly determined by how their host or vector species moves. We may ask if this movement can be detected in viral genetic data. Well, it all depends on the relative scales of movement, viral evolution, and sampling through time and space. If movement is too fast compared to evolution, then not enough mutations will be present to discriminate isolates from different locations. If sampling is too infrequent in time and space, then movement will be underestimated. Phylogeographic analysis helps us get insight into the pathogen's population structure. In the case of panmixis, isolates from different locations do not cluster phylogenetically, meaning that there is no population structure. This is observed in several human respiratory viruses like influenza or SARS-CoV-2, as the hosts may travel long distances before transmitting the pathogen. On the other hand, there are cases of strong population structure where isolates from the same location are typically phylogenetically clustered. This is often the case with sexually transmitted and blood-borne viruses that follow social or linguistic boundaries. Gravity model dynamics are often seen on highly transmissible human viruses. Here, transmission among locations is proportional to relative population sizes, such that epidemics spread among big cities first before disseminating to outlying villages. When a phylogeny shows a strong correlation between genetic and geographic distances, it reflects a wave-like or diffusion spread. This is often seen when transmission and/or spatial movement rates are low, and is more common for wildlife viruses than those of humans. Keep in mind that signal of spatial structure is lost if the epidemic changes direction. When we have two samples of a pathogen, we might infer an apparent direct movement when the real movement is more complex. This can be improved if intermediate locations have been sampled. One of the models that can be used to retrieve spatial information is the discrete model of spatial diffusion. To explain this, let's consider a set of samples with discrete locations. Similar to nucleotide substitution models, here we use a continuous time Markov chain process to model pairwise rates of location exchange, which are represented by parameters in the rate matrix. However, for a single location observation, this poses a challenge. We have to estimate a considerable number of rate parameters. Fortunately, Bayesian stochastic search variable selection, or BSSVS, allows us to do this in an efficient manner by reducing the number of parameters to estimate, which most likely explain diffusion and estimate support in terms of base factors for phylogeographic connections. Now, the aim is to jointly reconstruct the evolutionary history and the discrete location state at the ancestral nodes based on the sequence and location data observed at the tips of the tree. This kind of ancestral reconstruction has been used extensively in molecular epidemiology and has largely substituted older parsimony approaches. The first time this approach was used was to reconstruct the phylogeographic history of avian influenza H5N1. One of the most prominent settings when this model was applied was to identify the origin of the H1N1 pandemic, showing that it emerged from Mexico in 2009. However, not all sampling is discrete, and each sample can have instead latitude and longitude associated. Botanist Robert Brown, from where Brownian motion takes its name, identified and modeled the random motion of suspended particles caused by collisions between them and the medium molecules. We can use this rationale to explore a random walk in the coordinates throughout the tree to perform ancestral reconstruction. Employing a strict Brownian diffusion to model spatial movement makes the assumption that the process remains homogeneous over the entire phylogeny, such that the same rate of diffusion applies to all branches at all times and between any two places. To remedy this, the relaxed random walk borrows from the relaxed molecular clock models, such that the diffusion rate on each branch of the phylogeny is drawn independently from an underlying rate distribution. Since I'm going to present you several of these reconstructions, I will now show you how we can represent them. Both methods, the discrete and continuous models, estimate locations at the ancestral nodes, and we want to see the spread over time. Visualizing both time, space, and uncertainty simultaneously poses a challenge. Our approach goes by projecting the phylogenetic trees in a geographic space over time. In addition to visualizing, we can draw statistics that help understand spatial spread. This includes, for example, dispersal rate, wavefront, and diffusivity, also known as diffusion coefficient. The dispersal rate measures how fast the virus spreads per time unit. The wavefront is a measure of how fast the virus is spreading through time from the origin location. And the diffusivity is the area explored by the infected host.